let's take a little look-a-roosie, shall we? So like we said, one of the first things we need to make sure we're comfortable with is the idea that we're now going to be working with DNA, which we should be pretty comfortable with by now, deoxyribonucleic acid, right? And then we're also going to be working with RNA, ribonucleic acid. Differences: DNA, the sugar involved is deoxyribose. RNA, the sugar is ribose. The difference of those being ribose has one more OH group. DNA is double-stranded. RNA is single-stranded. Thymine. As in DNA, we have cytosine, guanine, adenine, and thymine as the 4 nitrogenous bases. Of those, which are the pyrimidines? The pyrimidines. Adenine and guanine are the same type, but they're not pyrimidines. They are purines. And so the pyrimidines are cytosine and thymine. In ribonucleic acid, we have the same purines, adenine and guanine. For pyrimidines, we have cytosine, but we also have uracil, which is very similar to thymine. It lacks this methyl group here, the CH3 group here. Other than that, similar. DNA is just used for information storage. That's its function. Its function is that it serves to store our genetic information, and it's so incredibly stable, it doesn't do much else. On the other hand, RNA is used for information storage, but it's also used as a catalyst. It's also used to perform functions in the cell. So RNA shows up in all kinds of cellular functions. So we said transcription at its most basic is this. Thank you. At its most basic is this. There'll be a section of the DNA that has a gene, which is to say a segment of the DNA that codes for an amino acid sequence, a polypeptide. And the cell isn't going to make an amino acid sequence directly from the DNA because we don't want to have to interact with the DNA over and over and over again to damage it. So instead, when there's a section of DNA that we want to turn into a protein, we want to turn into an amino acid sequence, what will happen is that a new molecule, a new enzyme that we haven't dealt with yet, which is called RNA polymerase, will come along, and it will work. We'll get a little replication fork. We'll open up the DNA just like we would in DNA replication, but RNA polymerase will bind, and as RNA polymerase binds, it will move across, and it will make a copy of the DNA, but not out of DNA, but out of RNA. Because RNA is single-stranded, that's not going to bind to the DNA. It's going to simply float off and be its own molecule. And then the original strand of DNA can simply be refolded back together. How's that feel? Yes?
So the point of the lesson 2 days ago was to replicate DNA. What's the point of this lesson? Is it to make proteins?
That's right. Yep, so the whole point here is that we're going to make protein, but the first step is actually that we make a copy of the DNA, but the copy of the DNA is made with mRNA, and M stands for messenger, so literally we call it that because this RNA is then going to leave the nucleus, because all the machinery to build proteins isn't found in the nucleus, it's found in the, according to here, cytoplasm, but I'm assuming that's supposed to say cytoplasm. Yeah, so now we want to look at this in a bit more detail, what this looks like. So we said the first stage is what we call initiation. And initiation, to initiate something simply means to start, to get started. So to get started, you got a lot of DNA. So how in the world, if RNA polymerase is going to copy just one gene, how does it know to do so? Don't just point. It's not helpful. I wrote it. I know it's there. How does RNA polymerase know where a gene starts and where it ends? It doesn't know because it doesn't have a brain. This is very true. We're using anthropomorphic language. But by what mechanism does it find and bind to the DNA in the right spot at the beginning of gene? Yeah, we have these regions on the DNA itself called promoters. And the promoters are just segments of DNA that have a specific code, and the RNA polymerase molecule is structured in such a way that it binds to those spaces. So we looked at bacteria are famous for TADAT and TGACA, these two sequences, these two promoter regions. In humans, we have what's called the TADA box, which is that 10 base pairs behind the actual beginning of the gene, we have this typical sequence, thymine, adenine, thymine, adenine, adenine, adenine. And that sequence is required for RNA polymerase to bind to the DNA molecule and begin transcribing. So that sequence appears in front of our genes. And then the gene itself will be on ahead, but this is the spot where RNA polymerase can bind. Now, interestingly, if all of our genes have this, that still doesn't really answer the question. So now we know how RNA polymerase knows how to find the beginning of a gene. Next question, how does it know how to find the beginning of the gene that you want copied? You know what I'm saying? So this would be the equivalent of saying, okay, if we are in a library, we can tell where one book stops and another book ends because we know what the cover of a book looks like, right? So we can be like, oh, look, between these two covers, that defines a book. That still doesn't tell us how, if you're going to the library and you want to get a specific book, how do you find the specific book? You know what I mean? So there's a lot that goes into that, gene control. But at its most basic, we want to talk about transcription factors. And the basic idea there is that just like RNA polymerase interacts with the promoter by being able to bind to it, you can have other sequences out in front of the promoter that different enzymes can bind to. So you can design an enzyme, or the cell has evolved enzymes that will bind out in front and stop RNA polymerase from binding there. So if there's a gene that the cell doesn't need to use right now, you could have an enzyme that locks on here, and then it will block RNA polymerase from binding to the promoter. And if RNA polymerase can't bind to the promoter, then transcription can't happen. Does that make sense? On the other hand, you could have another site called an activation site out in front that when a different enzyme binds there, it actually attracts the promoter. It says, like, there's a high affinity between them, which the result of which is equivalent to saying, hey, over here, this is the gene you want to copy. Does that make sense? It actually attracts the promoter. It attracts RNA polymerase not just to any promoter, but to this specific one here because it's next to this activation site that an enzyme can lock onto. And some genes have several activation sites, all of which have to be occupied. Some genes have several activation sites, any one of which can be occupied. Some genes have activation sites and inhibition sites. It depends on their function in the cell. Is that logical enough? In a few days, you'll learn about a specific one, which is how your cell regulates making an enzyme called lactase that will allow the sugar lactose to enter your metabolic pathways instead of glucose. And normally your cell doesn't bother making that enzyme because if it's got lots of glucose, you know what it doesn't need to metabolize? Lactose. But if the cell finds itself in a condition where it has a lot of lactose and doesn't have a lot of glucose, all of a sudden it will go turn that gene on and start producing the enzyme lactase, which is kind of cool, right? So your cell has a mechanism where it can say, hey, we've got a lot of this food supply. We should make the enzyme that will digest it. And we'll look at exactly this example to show you just for that one gene, how it's actually controlled by the cell. So the cell can say, turn this gene on now, turn it off now. And of course, we say this, control that. It's all a process that happens automatically by how these things have evolved and how they are function. Yeah.
That's the one that people who have lactose intolerance don't have yet.
That's right. Yeah, exactly right. Yeah.
What is this over there where it says like which prevents INP?
Yeah. So in this example, I've drawn a random example here, which is every gene has to have in front of it the promoter region, which for humans is what again? What we call the TADA box, right? This particular sequence. So a gene has to have that if the enzyme RNA polymerase is going to come bind here, go across and make an RNA transcription of that. But this particular gene also has an activator site where if the activator site gets bound to by an activator, then that will actually greatly increase the probability that RNA polymerase will bind here because it's designed to attract RNA polymerase. So under certain conditions, the cell can have an enzyme come bind at the activation site, which will attract RNA polymerase. On the other hand, it also has this section that we call the inhibitor, and the inhibitor, oftentimes it just physically blocks the promoter. It's just that an enzyme will bind to the inhibitor, and it will overlap and literally just physically cover the promoter region, in which case RNA polymerase just can't bind, and so it doesn't, and so now the gene won't get used. The gene won't get transcribed. So what it says over here is that enzymes hypothetically could bind to the inhibitor, and that would prevent RNA polymerase from attaching to the promoter. On the other hand, other enzymes could bind to the activator site, and that would actually attract RNA polymerase.
So in that example, most likely you would find RNA with that activator site.
Yeah, so it depends. Let's say this is a gene that, I don't know, it's something that we need to do sometime. Let's say like salivating, right? Like, so once your body like starts actively salivating, obviously you lose saliva as you go, right? Like the nature of it, right? Like it's coming out your mouth, you're swallowing it. So all of a sudden the glands in your mouth, if you start salivating, are going to have to start making new saliva. And let's just look at one enzyme. Let's say the enzyme amylase, which breaks down starch. Okay, so that's in your saliva. Now that I'm talking about salivating, I'm thinking about lemons, and now I'm salivating. Is anyone else salivating? Having heard this discussion, you can be honest. This is a safe space. You are, aren't you? Okay, that's okay. So anyway, so when that happens, those cells are going to have to start pumping out amylase, right? On the other hand, most of the time, they can do that really slowly. Does that make sense? They don't need to kind of pump out a lot of amylase because most of the time, the saliva that's in your mouth sticks around for a good amount of time, right? So I don't know anything about the gene that regulates saliva production, but what I would imagine, just knowing what I know about it, is probably that most of the time there's an inhibitor attached to that because most of the time there's no reason for the cells to be pumping saliva out, right? And then there's probably some signal that gets sent by our nervous system to those cells, and that's probably a hormone. And that hormone, the way most hormones work is the hormone isn't making its way into the nucleus. That's not, for protection reasons, that's not the usual mechanism. But there's something called signal transduction pathways, which basically means that, you know, a hormone would bind to those cells and say, hey, time to salivate, you know. And then when that happens, that would eventually lead to a signal molecule getting released into the nucleus whose job is to bind to the activator. And maybe that is designed that once it binds to the activator, it's designed to kick out the inhibitor. And once that happens, RNA polymerase can bind, and then it would go. Does that make sense? And then when that hormone goes away, when we don't need to be salivating anymore, then this molecule, the one that binds to the activator, would stop getting sent to the nucleus. And so eventually it will degrade and go away, at which point that inhibitor will be able to rebind here, at which point the promoter will be blocked, and now that cell will stop making amylase again. Does that make sense? Mechanisms like that are how the cell manages when to transcribe a gene and when to not. In other words, mechanisms like that are how a cell either initiates transcription or doesn't initiate transcription. How's that feel? Not too bad? Just bad enough? Okay. I do feel for some reason today vaguely like I'm torturing you. Am I torturing you? Charlie, don't hurt. Oh, that hurt my feeling. Okay. Well, so be it. All right. What comes after initiation? Elongation. All right. So what does that mean? Yeah. And specifically, what it means is that we're elongating the mRNA strand. All right. So in stage 2, RNA polymerase produces an RNA strand from the single-stranded DNA template. All right. So we have our template strand of DNA, and it works in the 5' to 3' direction. Does that look familiar? What else works in the 5' to 3' direction? Sorry, what? DNA polymerase, right? So RNA polymerase works exactly like DNA polymerase. The new strand is going to start with a 5' end available, and the last nucleotide will have a free 3', right? So let's look at this. We have our original DNA. All right, so here we are, and let's say this is the 5' end of the DNA, then this will be 3'. And then initiation has taken place. So we have our promoter region here maybe. What did I use for that? So we have the promoter there, and the promoter has attracted RNA polymerase. So this is going to open up like that. That did not go well. Okay, and what would this end here be? 3', good. And that would mean this one here would have to be 5'. Okay, so we're going to call this the template strand. And if that's the template, then it means that this section here of DNA, this section here is what's going to get copied. Does that make sense? And so RNA polymerase is going to bind, okay, just like we said, and it's going to move along. Now let's imagine that by the time we get to it, RNA polymerase is here. Does that make sense? So that is RNA polymerase. I'm going to draw that. It binds here, and it's going to move its way along, and we're going to imagine that at a certain point we get to here. So this is RNA polymerase. Well, RNA polymerase is going to be producing a complementary strand of mRNA the whole time that matches that. Does that make sense? So, well, it doesn't match the template strand. It should be complementary to the template strand. So, this is an mRNA strand. Hello. That is complementary to the template strand. Which means that this exposed end here, if this was 3', this will have to be 5'. And it's getting made, right? And the end here will be 3', right? And that's going to keep getting made as it goes along. Does that make sense? So this is made in the 5' to 3' direction. We've already said that, but we'll say it again. Yeah. Could you speak a little louder? I'm sorry. Good question. Okay, ready? So when RNA polymerase first binds here, it's going to make a complementary mRNA just for there. Does that make sense? So then, as RNA polymerase moves along, right, now RNA polymerase will be here, right? And by the time RNA polymerase gets here, maybe I'll show it as if it kind of hard to know how to, I guess I'll show it like this maybe. It will have covered that whole distance. Does that make sense? And that was a 5' here. So that has been made as it goes along. And that's the 3' end there, which for some reason I showed as a plus sign as if I was teaching a chemistry class, which I'm not. And I'll draw it a couple other ways as well in a moment, but maybe we'll make it a bit more clear. Yeah.
It's like a spider. Like the end of the, no, like.
Yes. Oh, yeah. As if it's like making a spider web.
The web, yeah.
Yeah.
One end is the 5' at the start, and then as it goes.
Yeah. Actually, that's if you want to think of RNA polymerase as a little spider walking across your DNA, and if you want to think of mRNA as its web, that's actually a pretty good analogy in that like the most recently added base pairs are here, right? Yeah, that's good. So, in this case, the RNA polymerase itself uncoils the DNA. And forms a complementary strand of mRNA. This is not attached, it's not bound to the DNA at all. So it's fundamentally different from DNA polymerase, because in DNA polymerase, as it's adding new base pairs, those are bound to the DNA, right? But the mRNA simply sits free. Does that make sense? By the way, if you think about it, the mRNA, if it's complementary to the template strand, then what does it actually end up being completely identical to? Yeah? It's going to be identical to this side here. So we call this the coding strand. We call it the coding strand because if you think about it, if you went and made a piece that was complementary to the gene you'd wanted, what you would actually end up with is the opposite of it, right? Whereas if you bind to and make a copy of its complementary pair, then what you get is the same as the code. It doesn't actually really matter which one you think of as coding and complementary, but just the sense that the mRNA interacts with one strand and ends up making a copy of the strand it actually didn't touch. Does that make sense? That's how the complementary nature of it works, right? And so I thought we'd draw that right now. So let's imagine we have the coding strand here, and let's say it's, I don't know, A, T, G, and I'm going to go C, T, A, G, C, T, A, T, T, G, C, T. Okay, whatever. That's the gene we want. Does that make sense? And then let's say that we have the complementary strand, what we call the template strand right here. So the template strand would be what? T, A, C, and then G, A, T, C, G, A, T, A, C, G, A. Okay, so now let's imagine that polymerase has made it this far. Does that make sense? So let's say that right now it's making the complementary pair to this A. Well, what normally goes across from A? T. Now, of course, because this is mRNA, mRNA doesn't have T. What takes its place? Uracil. So instead of a T across from an A, we'll have a U. Does that make sense? And then it'll start to put this one on, and what's going to go opposite the T? A. But the rest of these have already been copied, right? Because the RNA polymerase is moving this way. And unlike when we make a copy of DNA where it would be stuck to this, the rest of this will just be free floating. Does that make sense? So what would have been across from the G? C, and from that C? G, and then T would be A, and then A would be U, not T, good. G would be C. Across from C would be G. Across from A would be U again. Across from T would be A. Does that make sense? Yeah. Nope, it's all good. I'll show you. Because one of the differences between DNA and RNA is, and this is not a huge difference, but if you look, the fourth nucleotide in DNA is called thymine, and in RNA, it doesn't have thymine. It has a very similar molecule. The only difference is it's missing this CH3 group here, but RNA, in lieu of thymine, has this molecule, uracil. Because it's complementary, right? So just like DNA is complementary, A and T are always together. But now the other thing you have to know is that A and T are always together, but that also means that A and U are always together. And then G and C are together, C and G are together, T and A are together, A and U are together, G and C, C and G, A and U, T and A. Yeah. It just gets read as T. In other words, keep in mind, it is almost identical to T, right? It's got that one little difference. It fulfills the exact same purpose. It just happens to be different. And why are they different? The answer is that one evolved from the other. We understand DNA to have evolved from RNA, and in the process of doing so, it evolved to have a difference. But functionally, it'll do the same job. So the truth is, all the machinery that actually builds proteins does so using RNA. And so it expects to see a uracil. The machinery that stores the information long-term in our DNA uses DNA, so it has thymine, and the machine that transcribes from one to the other knows that where on the DNA a thymine would go, it's going to put a uracil. Of course, it doesn't know that. That implies the sentience that isn't there, but it has evolved to pair things off in that way. Did that answer your question at all? Okay. Hey, look at that. So this is the mRNA molecule. What's the M stand for again? Messenger. Very good. And it's worth noting that it is, it is free from the DNA, right? I.e., it's not bound to it. The only reason it's not floating away completely is it's not free of the actual enzyme. What is this blue splotch I've drawn? That's the RNA polymerase. And that is the only thing that the mRNA is stuck to. Now, these parts that have already been read, they will come back together. Does that make sense? These will reattach, and the DNA will go back to just being what it is. And keep in mind, of course, this section will unattach as the RNA polymerase works its way across. That make sense? But what I want to make a note of is if we compare this sequence to that sequence, what do we see? It's the same. The only difference is where in the coding strand of DNA there would be a T, in the mRNA strand there will be a U. I think that highlighting actually is too much. I think it makes it hard to follow, so I'm going to get rid of it. But what I am going to do is make a little note and say that the mRNA sequence is complementary to the template strand. Which means it has the same sequence as the coding strand. Except for what one difference? Except U in lieu of T. And again, what does U stand for? uracil. All right. And again, just to quickly kind of look at this in a slightly more schematic way, the other way we can kind of look at that is we start off with DNA, right? And maybe this section of it here is the gene we want to make a copy of, right? So then what will happen is that RNA polymerase will come along to open that up, right? And maybe that's the gene we want to make a copy of, right? And if that's the gene that's actually coding, then where's the RNA polymerase actually going to bind? It's actually going to bind to the opposite side, right? If that's the gene we want, if that's the code of the gene, then RNA polymerase is going to bind here, right? And it's going to work its way down, and it's going to start making an mRNA copy, right? Until what we end up with is, so give me just a second to get this incredibly crappy drawing done. I'm so ashamed right now. You know what? I'm doing my best. It's the best I can do. This DNA looks as though it has suffered horribly, but that's okay. So it's working its way down, and by now we have a long sequence of mRNA, right? And then the DNA will be coiled again, returned back to, hey, that's pretty good, right? That one's pretty good. That solid C+. Okay. Cool.
What are you doing today?
I'm not sure. Come pop by and see. If I'm free, I'm all yours. I haven't checked my emails yet, so I may well be busy. You had a question. Was it, how did you get so good at art? Born that way, baby.
I answered it. I got the answer. But I was just going to ask, was it RNA?
Yes. Yeah. Yep. The RNA polymerase. In fact, I'm not drawing it this way. The RNA polymerase is actually a really big enzyme. So it actually really kind of like really sits on top of the DNA. Just to make this sort of schematically visible, I'm kind of drawing it as a small little guy. But it's a big, gives that DNA a big old bear hug. And then, as bears do, rips it apart, dismembering it. Because bears, godless killing machines. Hence, why they're on notice. Yes, ma'am. What are the numbers? Okay. 5' and 3, which again is because the mRNA, when it starts getting made, we get the 5' end here, and the last nucleotide that gets put on the mRNA will have a free 3' end. So the first nucleotide has a free 5' end, the very last one has a free 3' end, which means that that's actually true of, if we follow these along, if we just look at the gene itself, that's the 5' end of the gene, and that's the 3' end of the gene. That's the 3' end of the template strand, and that is the 5' end of the template strand. I'm not going to put all those there because I think it makes it more confusing, not less. But just like these two strands go opposite each other, this strand will be the opposite of the template strand, which means it'll actually be the same as the coding strand. Again, it's interesting that the mRNA never interacts with the coding strand, but it ends up being functionally identical to the coding strand, even though the strand it interacted with is actually the one across from that, the template strand. Yeah? In lieu. Lieu. Does everyone know that word? Lieu? Lieu just means like in place of or instead of. You in lieu of tea. That would be funny if you didn't get into U of T and then you went to a different university and you referred to it all the time as Lou of T. In lieu of T, where do you go? Lieu of T. So you say it quick, like if that was important to you. For the record, it shouldn't be important to you because every school in Canada is a good school. But if you have an ego about these things. What I've discovered, my long years of experience of knowing people who went to U of T is it's very important to them that you know that they went to U of T. I've known a few people in my life who went to like Ivy League schools. And anyone who went to an Ivy League school, if you're ever wondering, how do I know? You'll know because it will come up in the first 10 minutes you ever talk to them and every 10 minutes thereafter. People who went to fancy schools will always find a way to mention something that happened at their fancy school. No. I think...
Like Greg or his friend Greg? Like
my son Greg. But he'll probably build up to it, I'm sure.
He's a little confusing.
Yeah.
Wait, no, his friend Greg went to school with him.
That's true. So, my friend Greg did his undergrad at Brock, and then he did his master's at Western, and then did his PhD at the University of Ottawa. So, he got around. Yeah. Yeah, if you do science sort of academically, you get bounced around at first. That's like anything else. And then you sort of tend to settle in somewhere. But you can't be a university professor, research scientist just anywhere. You're going to have to go to where there are universities and then where they're hiring, right? So something to keep in mind. Sorry? He has a lab at a university, so he's not a teaching professor. So he is a professor at the University of Ottawa, but he actually works for Health Canada and does research for them. My friend Greg would be an incredibly awkward... I would love to see that. I'd love to watch him teach. He has taught before, but it is not his passion, shall we say. He doesn't like... He's not the most... Ironically, not the most gregarious person in the world. I always ask him if he wants to come do a session for like one of my bio classes, like on physiology, and he always says no. He's a shy guy. Anyway, I wish I were a shy guy. All right. Now, obviously, this happens until we get to the end of the gene. So the truth is, this is actually the next step. This is after the final step, which is termination. And what does it mean to terminate? No. I mean, I guess, euphemistically, but really it just means to end, to stop, to finish. Which I guess, yeah, if you end somebody, one of the ways you can do that is by...
Terminal illness.
Boy, guys, you guys are dark. What about like the bus terminal, which is where like the bus stops? Like we could... Right? Yeah. Right?
What terminal?
Terminal velocity, which is when you're skydiving and that's as fast as your body can go because even though gravity is accelerating you, wind resistance is accelerating you in the opposite direction. Yeah. Physics. Has anyone here been skydiving? I have not been skydiving. Should I?
Class trip, guys.
I'm a coward. So it's very important for you to understand that I'm a deeply fearful person. And two things can be true. You can be tough and a coward. Part of being a coward is... I will... Also, it's out there. I think most people, actually, who like put on big shows of toughness are just covering for deep insecurities and cowardice, actually, would be my hot theory. Tell me if you disagree. Anyway, regardless, certainly that's true in my case, and let's move on. So, I might just be projecting. So, at the end, what has to happen? Well, at some point, it has to stop copying. Otherwise, it would just make a copy of the whole rest of the chromosome, right? Which, the whole point of this is we want to copy just one gene, right? So, on the DNA strand itself, again, there's a sequence of base pairs that are what we call the termination sequence. So just like we had the promoter region, which promotes RNA polymerase to bind, we have the termination sequence, which tells RNA polymerase, all right, get out of here. Again, that's obviously anthropomorphizing it. There's no speaking involved, nor Italian grandmother-style hand motions, but you get the idea. On the DNA strand is a sequence of base pairs. And I'm going to use this abbreviation BP for base pairs because you're going to come across that a million times if you look up any bioinformatics. this causes RNA polymerase. Is everyone comfortable with me just calling that RNAP to cease transcription? I say I would write that, but I would never say that. I would say RNA polymerase. If you work in education, you will quickly discover that you hate acronyms with just an ever-loving passion. Calvert finds them funny. He always refers to TLAs, all the three-letter acronyms in teaching, which is pretty funny. Yes?
Is the termination sequence on the DNA strand that causes RNA transcription the last strand of the DNA?
Does it actually, are you asking, does it itself also get transcribed? I'm not sure, actually. That's a...
I'm asking, is the DNA end where the RNA sees?
No. Because keep in mind, a gene could be anywhere on the chromosome, right? So the DNA keeps going. So the termination sequence is the portion of the DNA that says, hey, you can stop copying now, but the DNA strand keeps going. That was a great question, by the way. Yeah, very important to understand that you have hundreds of thousands of genes. So this is where it says, okay, stop copying, you're at the end of this gene. And then RNA polymerase can unbind. And then, of course, the DNA will have to return to its normal configuration. And we have a term for this. Has anyone ever gone down the rabbit hole of watching like blacksmithing videos on YouTube? Oh, guys, you're missing out. Would recommend. Okay, welding's cool too. But these guys who have like forges set up in their backyard, and they're like, I'm going to make a blade of Damascus steel today with my... Yeah, see? Oh, you didn't want to admit it, but you've watched a guy in his backyard with a power forge, or with a power hammer, forge a Damascus steel sword. Yeah, me too. Because it's fascinating and cool. And then they always have the bit at the end where they like cut stuff, and you're like, that looks like a lot of fun. I wish I were doing that right now. But anyway, do you know they do the step where they anneal the steel, the metal? Have you heard of that before? You don't know what that means? It's actually, sometimes they also just call it quenching, where they will actually heat the blade back up and then cool it really quickly. Does anyone know what that does? If you cool it really quickly, what you can do is, if you cool it slowly, that is called annealing. And if you cool it slowly, while it cools, the grains of the metal will naturally realign themselves up, and you actually end up with a softer metal, which sometimes you want. But sometimes if you want the metal to be very hard, you want to cool it quickly so that the molecules don't have time to, or the metal atoms themselves don't have time to realign themselves and form that pattern, and you can get a more rigid structure. And so that's how you cool the metal determines how hard it will be in metalwork, which is really cool. But they call that process of slow cooling, which allows it to form back to its sort of natural inclination shape, annealing it, versus quenching, where they do this quickly. That has nothing to do with this, except we share the term. We talk about DNA being annealed or re-annealed, which means taking on its normal shape, letting it return to its normal double helix shape. So the DNA will be fully re-annealed. In other words, recoiled into a double helix. And the more complex fibers. And when I say the more complex fibers, you remember that from the beginning of the previous note, right? How they wrap around the histones, forming the nucleosomes, how those fold, and you get the 30 nanometer fiber, blah, 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 blah. So the DNA will be fully re-annealed, and the mRNA strand will be free. I do want to make a note, just that in reality, genes are very long, and sometimes there could be 2, 3, 4, 5, there could be a bunch of RNA polymerase molecules all transcribing the same gene at the same time at different locations. Does that make sense? really popular genes, genes that the cell has to use a lot, might get transcribed a lot so that we can make a lot of those proteins. And obviously, genes that are used less frequently will be transcribed less frequently. Yeah. Is it related to the which? One more time, nice and loud. Is the termination sequence related to the inhibitor site? No. But it's the same idea, which is the inhibitor site mostly prevents transcription actually by just physically blocking the promoter region. There might be other mechanisms, but certainly when I learned about it, most of the mechanisms were literally that you have the inhibitor site and then the enzyme that binds to that is just this big bulky thing such that it literally just covers up the promoter, and that way RNA polymerase just can't find the promoter and so it can't bind. Whereas the termination sequence is actually more closely related to the promoter sequence, where the promoter sequence attracts RNA polymerase, which makes it bind to the mRNA. The termination sequence actually repels mRNA. mRNA its active site is such that when it hits that specific sequence, it bounces off of the strand. Did that answer your question at all? Okay. Note that more than one RNA polymerase molecule may be transcribing a given gene at any given time. These days have gone, but when I went to university, we still had to get, if they had an article they want us to read, they couldn't post it online, so they would post it on reserve in the library, where there'd be 5 copies of this article at the reserve desk of the library, and you'd go there and you could sign it out for 10 minutes, which obviously wasn't enough time to read it, but it was enough time to take it to the photocopy room, make a photocopy of it, and take that with you. My God, the amount of time spent in the photocopy room when we were in the university. But to save time, I'd take one reserve out, you'd get a friend to go get another reserve out, because often you'd be waiting, right? There's 10 copies on reserve, 1,000 people in the class. So you'd wait there and wait till somebody returned one, and then you'd snag it. And while you're copying that, you'd have your friend sit there and wait for the other article on reserve, and when he'd get that one, he'd come over and start copying it. And you can imagine this kind of chaotic sequence where you're half done copying yours and passing it off to your friend, and they're half done copying theirs and passing it on to you. That's kind of what this can be like, where if there's a gene that we need to transcribe a lot, one mRNA can be working right at the beginning of the gene, one might be halfway through, one might be near the end, and they can all be transcribing the same gene at the same time. Does that make sense? Let me tell you, nothing is lost in the era of having, some things are just to the good, and it is to the good that you will never have to have your stupid library photocopy card and be waiting. My hateful, spiteful relations with photocopiers started young. Specifically, those first-year classes, trying to copy, you've had a history thing, because they would charge you money. For every minute your reserve was back late, you'd get a late fee, because they obviously can't have people keeping them out. And so I'd have the photocopier jamming and be like, oh my God, I need to get this back. I'm glad that you never have to do that.
How can they make it later? They just need to upgrade their machinery.
That's how life works.
Okay, yeah, but
That's just how life. No, it's just how life works. It's just you gotta get back on time. It's just how life works. But the counterpoint to that is, why should somebody else have to wait because you screwed up using the photocopy? You know what I mean? It's just how life works. You gotta get back on time. So if you couldn't get copied, you go take it back. In Paris, they have what they call the Velobond, which are these bikes that you can sign out, and they're quite cheap. For $1, you can have a bike for an hour. But then every minute you're late is quite expensive. And so the system is they don't want people renting these bikes and then having them all day, because then they're not available to someone else. But you can return it to any place. You don't have to return it to where you got it. You can return it to any of the bike drop-off locations. And so if you're out biking around and you've got 6 minutes to go, you find another place, return yours, sign another one out for a buck, and it's kind of no big deal, right? But that system presents somebody paying $1 and then having a bike for the next 18 hours or 20 hours, or just stealing it or whatever, right? So arbitrary systems may seem arbitrary when you're the one being inconvenienced, but when you zoom out and look at the system in general, that you might get inconvenienced 5% of the time is unfortunate, But if it makes the system run smoothly 95% of the time, well, it's a good system, right? It's part of thinking of others, right? Part of thinking of others means that the system won't always be most optimally convenient for you, and we have to be at peace with that because we live in a civil society, right? And, I'm at peace with that, but it did foment my hatred of photocopiers. Oh, yes, it did. All right, now, you'll recall that we said that the central dogma is that one gene codes for one polypeptide, one amino acid sequence. So there's a few things to know. The first thing to know is that DNA transcription is not always, or even often, of a single gene at a time. The mRNA that is produced from the DNA template is typically modified before it's moved to the cytoplasm to be turned into a protein. And we're going to talk about a few modifications, just a few. The first is what we call the 5' let's try that again. The 5' cap and the poly-A tail. As it sounds, the poly-A tail is that on the 3' end, there's just a sequence of A's added. So a long sequence of A's, adenines, is added to the end, to the 3' end of the mRNA. This serves as a form of protection because as it turns out, the nucleotides most likely to be damaged are on the ends. So if you add a big chain of gobbledygook at the end that doesn't matter, if it gets damaged, it's specifically adenine. Yes, protection. If this sequence is damaged, no harm, no foul. The message is unaffected. The other thing that happens is that we get this 7-methylguanosine cap. Basically, on the phosphate of the first nucleotide, in other words, on the 5' end, this 7-methylguanosine cap is added, and this prevents the RNA from being attacked by catabolic enzymes. Basically, when the mRNA is done with later on, there's going to be enzymes that just break it apart so that the nucleotides can be reused, right? Recycled. And this cap says, hey, I haven't been used yet, don't recycle me. Does that make sense? So it serves as a signal. It protects the RNA from catabolic enzymes in the cytoplasm. So this is attached to the 5' end. And it prevents, I should say protects. That's the language I use. I'm going to say it protects the RNA from catabolic enzymes and this serves to initiate translation, which is our next lesson, which will be a couple days from now, in the cytoplasm. So the next step where this actually gets turned into a protein, the enzyme responsible for doing that, or the mechanism responsible for doing that, it is initiated by this 7-methylguanosine cap on the 5' end. This is next lesson. What is catabolism? Break it apart. And again, what that means is when your mRNA is finished, like once we've turned it into a protein, we don't want to destroy it, but we also don't want it floating around. What we want to do is reuse all those nucleotides. So as soon as this mRNA has been used to actually make a protein, then a bunch of enzymes are going to come and just rip it apart. And then we'll reuse all of those nucleotides. But obviously that would be inefficient. It would be problematic if that happened before initiation. So as long as the 5' cap is there, the catabolic enzymes don't break it down, and that 5' cap is also what starts translation. During translation, the 5' cap gets removed, potentially, and that means that after translation, which is, again, the next part where we actually make a protein, then the catabolic enzymes can rip that apart so we can recycle the parts. Yeah? It's going to go to the cytoplasm, and there it's going to get turned into, it will be read as a template to build a protein. Right? Because again, what this actually codes for is an amino acid sequence. All we've done right now is make a copy of the DNA, so we still haven't actually used it to make something. What we've done is we've made a photocopy, like I used to do in the library, as what was on reserve, so that then I could go to the cafeteria, or to a friend's storeroom, or to one of the quiet spaces in the library, or to my own bedroom at home, and do the actual work on it. Does that make sense? In this case, we make an mRNA copy of the DNA so that we can send it out of the nucleus into the cytoplasm, and the cytoplasm has all the machinery to read that and actually build a protein. But we don't want to put our DNA in the cytoplasm to build a protein because it could be damaged. So a copy is made, that is mRNA, that gets sent to the cytoplasm, and in our next lesson, we'll look at how that then gets read to make a protein. Did that answer your question? Amazing. Now there is one more thing, because there's always one more thing. Introns and exons. What the hell does that mean? Okay, well, basically, what we end up with is this. We end up with an mRNA sequence, and it looks like this. Okay, there it is. And here we have the 5' cap. Okay, and that means this is the 3' end, which also means that this will be a big sequence of A's, because what's on the 3' end? The poly A tail. Okay. And then this segment here, what we actually find...
Wait, is that cup or cap or nap?
Cap. Yeah, we have the 5' cap. So now, it turns out that in this segment, we will have little segments of this that, depending on what it is being used for, some sections will get cut out and some sections will be kept. Some sections will be cut out and some sections will be kept. And remember when we said that, like, naming in science can sometimes be really terrible?
Yes.
Oh boy, are we about to be in for one.
Do introns like leave and they like don't act? Oh, what the frick?
Yeah, listen, I didn't name them, all right? So we call these exons. Sorry? Okay, like are we gonna like take our tiki torches and like just settle down? All right. I mean, A, they're probably dead, and B, like, settle down. Okay, so some of the segments we refer to as introns and exons, and then what we have out here is we have that this segment here will be spliced out.
Mr. Bagnulo, please call the office.
By something called SNRNP, which we sometimes just refer to as, and I love this name actually, the spliceosome complex.
Oh, okay.
Right? The spliceosome complex. All right? So SNRNP is also known as the spliceosome complex. It is going to cut this whole section out. Right? It's going to cut the whole section out, and then it's going to splice back in. It's going to splice back in which ones? The exons. And it's going to leave out the introns. I don't know why. I don't know why that's how they call it. Isn't that, like, it's wild, right? Every time I teach this, I double check. I go double check the textbook, and I'm like, I have this right? And I do. Yeah, I don't know. Don't know what to tell you. But so splicing occurs, and what happens is that after splicing, we have the 5' cap over here, and we have that, and then we have the introns, All right. The answer is that the same thing can be spliced a few different ways. So depending on which protein is actually being made, they will cut out different introns and exons. And so you can have one gene that can actually give you a few different polypeptide sequences, which is pretty interesting. So it's basically efficiency. It's kind of the equivalent of saying, like, okay, take this blueprint. I have a thing. You ever bought a thing and it comes with an instruction manual, but the instruction manual is actually for like 5 different models, and it has the explanations inside for all 5, and you have to figure out like which one of these stupid ones, which one of this is my printer. It's kind of like that. This gene could actually be used. This gene could be used to do a few different proteins depending on how it gets spliced by SNRNP, also known as the spliceosome. And then the introns get removed and degraded. So the exons remain in the sequence.
Does that make sense?
So the exons get spliced back in. And the introns are removed and degraded, i.e. they get treated with those catabolic enzymes we talked about. So the introns removed and broken down.
Why don't you make it, Toby? How is it being like the freaking forensic, whatever you want him to be?
Okay, and then a note, a gene can have several, so it can have multiple intron-exon combinations. This comes back to that thing we talked about, epigenetics. Which set of introns and exons is spliced out has to do with what the cell is trying to produce, which is, as you can imagine, highly complicated. So intron-exon combinations, and therefore, it's actually the case that one gene can potentially have several different polypeptide sequences. So the one gene, one polypeptide sequence is the central dogma, but it's a little bit naive. The truth is that once we splice it, there can actually be a few different combinations. But now this whole bit here will be sent. This is sent to the cytoplasm. To form a protein. Right, so the whole sequence will then get sent to the cytoplasm, wherein it will be translated. And translated is the process by which we read the mRNA sequence and we actually build a protein from it. Does that make sense? Yeah. The whole thing. So the poly A tail is